Hi everyone, welcome to my channel Pure Biology, and today I come with another topic, that is the structure of muscle, molecular structure of muscle. Okay, I before going to this topic, I want to say thank you to all of you. Okay, let's continue. The molecular structure of muscle, the skeletal muscles that are attached to the bone by the tendons, and these are composed of one kind of connective tissues. Okay, this connective tissue, these are generally ensheaths the entire muscle. Okay, this connective tissue ensheaths the entire muscle and known as epimysium. What is called that? This is called the epimysium. And the skeletal muscle generally consists of numerous bundles. These are called the fascicles. Okay, the skeletal muscles, these are consist of fascicles, and each fasciculus, this is surrounded by a layer of connective tissue, and this is called the perimysium. So what is called the epimysium? Epimysium is the connective tissue that ensheaths entire muscle cell. This is called the epimysium. And in case of perimysium, this is generally this help to cover the one fasciculus. Okay, and which is called the fascicles? That means, sorry, and what are called the fascicle? Fascicle actually the skeletal muscle that consists of numerous bundles. Okay, and these numerous bundles are nothing but the fascicles. Okay, and each fasciculus, this is composed of numerous muscle fibers. Okay, one fasciculus, this is composed of numerous muscle fibers, and each individual muscle cell, this is surrounded by the connective tissue. This is called endomysium. So here the endomysium is which one? First of all, this one fasciculus, this is composed of fibers, muscle fibers, and each individual muscle cell, this is surrounded by endomysium. Okay, muscle fibers. So here the muscle only, not the muscle cells, the muscle that is covered by the epimysium, and fascicle that is covered by the perimysium, and here the muscle fiber that is covered by the endomysium. And the muscle fibers, these are very long and cylindrical striated fibers. Striated, unstriated means, striated means there are the band, that means stripe bands present, and unstriated means no stripe bands there. So muscle fibers are long cylindrical striated fibers, and generally they have many nuclei that located in the periphery. Sorry, one second. So there are many nuclei that are present in the periphery of the fiber. The cell membrane of the muscle fiber, this is called the sarcolemma. What is called sarcolemma? The cell membrane of the muscle fiber, this is called the sarcolemma. And the sarcolemma, this is specialized cytoplasm that is generally present in the form of the cytoplasm. What is called cytoplasm? That is present in the cell, any cell. But in case of muscle, there is the cytoplasm present is called the sarcoplasm, and the membrane, that is the cell membrane that present when in the muscle cell, this is called the sarcolemma. Clear? Next, one endoplasmic reticulum like structure also present in the muscle cell. This is called the sarcoplasmic reticulum. Next one is that transverse tubules. These are T tubules. These are invaginate the sarcolemma. That means these are present in the sarcolemma or the region of the sarcolemma. What is the function of that? They try to allow the impulses to penetrate the cell and activate the sarcoplasmic reticulum. Clear? Generally, they allow the impulses to enter the cell and activate the sarcoplasmic reticulum. It's the function of the transverse tubules. Next, sarcoplasm that contains thread-like, fine thread-like contractile structure. These are called the myofibrils. Each myofibril that actin thin filaments and myosin thick filaments, these are organized into a linear chain and called the sarcomere. So what is saying that within each myofibril, actin and myosin filaments are there, okay, as thin or thick filaments, and then this is called the sarcomeres. This is the unit of myofibril. Next, what is that? One end of the thin filament is attached at their plus end of Z disc, and the demarcation between the adjacent sarcomeres. So, generally the plus end, one end of the thin filaments, so there are thin and thick filaments present in the sarcomere. So, thin filament is attached with the Z disc where at the plus end, and there the alpha actinin is one kind of protein, and that anchors the actin to the Z disc and stable the actin filament. In that way, actin filaments may remain stable, because here the alpha actinin is one kind of protein that helps to bind the actin filament with the Z disc. Now the capped minus, that ends of actin filaments minus end extend towards the middle of the sarcomere where they overlap with the thick filaments. So the plus end is attached with the Z disc, and minus end that extended where they overlap with the thick filaments. They extended towards a region where they overlap with the thick filaments. Now, the thin filaments that contains, there are many proteins: actin, nebulin, tropomodulin, tropomyosin, troponin. And tropomyosin is very important for the knowing of the function of muscle contraction, or how the actin and myosin filament they work in the muscle contraction process. So here the tropomyosin and troponin, both proteins are very important. And what are the tropomyosin and troponin? Tropomyosin is the fibrous molecule, and alpha and beta chains are there that attach to the F actin in the groove between the filaments. Or you just know that this tropomyosin attached with the F actin with the help of alpha beta chain. Okay. Troponin, this is a complex of 3 polypeptides. What are they? That troponin T, I and C. What is this troponin T? Its function is seen in binding process, and inhibitory process is the I, and C is the calcium binding process. That means what is saying that troponin T's activity is seen in the tropomyosin binding process. Troponin I that has the function in the inhibitory process, and troponin C that has the function that calcium binding process. Clear? Now thick filaments, these are composed of what? 100 of myosin molecules. These thick filaments remain between where the Z disc, and there are another protein present. These are called the titin or titin, you can say. So what is that? Titin may have role in relaxation. So titin is a protein that may have role, or they show their role in the relaxation process. Now bipolar thick filaments that work generally towards the plus ends of two sets of thin filaments that opposite orientation and driven by the myosin heads. So what is saying that generally bipolar thick filaments that work towards the plus end with the help of what? With the help of the heads of the myosin. And they are positioned, these myosin heads, they position or they remain to the interact with the each thin filament, or myosin, they interacted with each thin filament. Clear? And sarcomere shortening also caused by the myosin filament. By what is the process by which these sarcomere shortening is seen? This is occurred by the myosin sliding past process. Myosin sliding past process. Okay. So these are the molecular structure of myosin. Another important part also there. So there is a space is very less, so I have to write after just rubbing it. So continue with this chapter. Okay. So the thick and thin filaments, they form a pattern alternating with each other, alternating with the light and the dark bands. So they form alternating way, light and dark bands. The light bands generally are called the isotropic or I band, and the dark band, these are called the anisotropic or the A band. So light band that containing only the thin filaments, and the dark band, these containing both thick and thin filaments. Now, there is a narrow H zone that remain in the center of the A band, and this containing only the thick filaments. Okay, some proteins are there, that supporting proteins that are called the supporting proteins. These hold the thick filaments together, and at the center of H zone form the M line. Clear? So, what is the role of these supporting proteins? These try to hold the thick filaments together where? At the center of the H zone, and in that way form M line. Why this is called the M? Because it is just in the middle of the sarcomere. So, this is the actual molecular structure of muscle, and in my next video I will try to explain about the muscle contraction process. Okay, so you just continue with my channel. Okay, and thank you so much.